This is the point where really if you're, let's say an Avengers fan or a Breaking Bad fan or something like that, this is where you have that final couple of episodes or that final movie in the sequence where all the ancillary characters come back and make their cameos. Things you thought were dead come back and now they matter again. You're going to see a decent bit of that here. The goal of this is that Chapter 12, while both preparing you to move on into physiology and to really be able to have a good understanding of how the metabolic processes take place in your cells, is also sort of built as a review of everything we've done. You're going to see a little bit of dealing with what form does this thing take under physiological pH. You're going to see enzymes involved. You're going to see feedback loops. You're going to see multi-step sequences. You're going to see a lot of the reactions that we've been dealing with all semester long as we take sugar and bit by bit turn it to CO2 and a whole boatload of ATP. Now, the worksheet packet for this week is a 10-page packet, mainly because I'm trying to turn this into your metabolism study guide for you as you go along. So one of the things this has right at the beginning are a handful of questions that are worth answering just so that we can answer this very rapidly as we go through the chemical reactions involved in glycolysis and the citric acid cycle and in fat metabolism. One of the things we want to do is remind ourselves, what are the things we make besides the organic products when we condense something or when we oxidize something? What are we usually consuming when we hydrolyze or when we reduce? Take a moment, pause the video, fill those things in, and when you come back, we'll check your answers. Okay? When we condense two molecules, remember what we're usually doing is removing an H from one side and an OH from the other. So for instance, when we're doing a condensation to put together a protein, for example, we wind up removing this H and that OH, and we turn it into an amide. So the other product we generate is H2O liquid. And by the same token, if I want to go the other way and I want to hydrolyze, let's say an ester linkage, which we'll have to do in order to break down fats into glycerol and the fatty acids. In that case, I'm going to break this bond right here. I'm going to stick an H on that side and an OH on that side and make my new products. So again, I'm going to be consuming liquid water in that case. By contrast, most of the oxidation reactions we've encountered in this class involved the removal of two H's. And notice I'm going to write it this way because in the human body, certainly if I'm doing this in a industrial plant, this could be what I'm actually adding. But in the human body, I'm not going to be burping or moving through my bloodstream hydrogen gas. I'm going to have H atoms that, as we've seen in the last video of last week, are going to get put onto or taken off of coenzymes. And so just reminding ourselves right here, that's going to be what holds these things, coenzymes, things like NAD+ or FAD. Okay? So just a good reminder for us. Now, the rest of this packet is going to guide us through a few steps, and you're going to see a lot of spots that look like this, where I've given you space for reactants and products. And I want to really emphasize when we do this, I'm not looking for you to draw the structures of the sugars, because it really, as far as that goes, the sugars are bit by bit going to get taken apart and turn into carbon dioxide once we're done. What I'm looking more at are the other reactants and products, things like water, things like coenzymes, things like ATP, and eventually things like CO2 when they get peeled off. Because what we're going to do by the end of this is sum up everything that happens in every step of these processes and give ourselves a lovely little summary reaction and see what it is that we've been doing this whole time. We're going to begin looking at glycolysis. And glycolysis is the first phase of metabolizing glucose. Remember from last time, polysaccharides, disaccharides, other monosaccharides, all those things have already been broken down and transformed into glucose or fructose by this point. Now, worth noting, excuse me, that fructose is going to have a place that it can plug in to this. So fructose and glucose are actually very similar to each other in terms of their chirality. If you look at everything past the ketone in fructose, it's identical to glucose. And so it's very easy for a fructose molecule to plug itself in here. In fact, you're going to see that rather soon the glucose molecule becomes a fructose molecule in this process. Now, the general idea of glycolysis, as it says on your worksheet, is, if you even break down the word, is just like hemolysis was blood break, this is sugar break. What we're going to be doing is breaking the glucose molecule into two halves. Two 3-carbon substances, so that we can start working on those a little more effectively and break the individual carbons off and oxidize them completely. So to do that breakage, what we're going to have are two general stages of glycolysis. First, we're going to tack on phosphates to activate. This might actually remind you a little bit, if you think about the phosphate idea here, of the way that the DNA backbone is spring-loaded, or the way that ATP is such a high energy molecule, is because of the presence of multiple negatively charged things in very close proximity. Bring two things that are repulsive towards each other, they repel, they're going to make it a high energy state. Okay, so that's the first thing we're going to do, is activate by adding phosphates, and then eventually that's going to cause a rupture of the molecule. We're going to break the glucose into two smaller fragments, and then we're going to remove the phosphates and make some ATP. The final organic product of glycolysis is something called pyruvate. And if you look at the ending of that, ATE, that should look familiar from when we did nomenclature earlier in this class. Pyruvate is an anion. It's actually the conjugate base of pyruvic acid. That's the thing that with one extra change is going to enter what's called the common catabolic pathway. And the common catabolic pathway is the one that the products of this process and of oxidizing and breaking up fat molecules can actually begin at. So everything from that point forward is the same, regardless of whether you began as a sugar or as a fatty acid. As I said, keep an eye out for the other reactants being used and the products that we make along the way. We're going to put them onto this worksheet as we go, and they'll be here on the slides. So first and foremost, reaction 1 of glycolysis involves us attaching a phosphate ion to a sugar. And if I wanted to connect a phosphate and an alcohol, if I want to connect any two biomolecules, what's my method? Yell it at the screen. Hope you've come up with the reaction of condensation. And so that's exactly what we're going to do here. We're actually going to remove a phosphate from ATP and attach it to this thing. So this first step, as we said, is activation. It's going to cost us some energy. Worth noting that if we are doing condensation, what did you just tell me? You're going to generate as well. Take a look at the top of your worksheet. Hope you came up with water. So that is definitely being generated here. And if you're working through the worksheets as we go, here's what this might look like. So we know here reaction 1 is a condensation. And what we just saw here is that one of the products of this is going to be H2O. It's also worth noting that the other thing we did is we consumed a molecule of ATP and made a molecule of ADP as a result. We turned it into that. So those right now are our new reactants and products, along with the glucose and the glucose 6-phosphate. Okay? Now, reaction 2 is actually nothing more than a rearrangement, technically an isomerization. So remember, isomer means that we have the same chemical formula but a different structure. And here I was just alluding to the fact that glucose can rather easily become fructose by way of a rearrangement. In fact, it's the same rearrangement that takes place and is responsible for the fact that fructose is a reducing sugar. The fact that glucose and fructose can easily interconvert by moving where that double bond is located means that when I have fructose, it can slide over and become glucose, and that allows it to go through the reduction, or excuse me, the oxidation process with that aldehyde. And by the same token, glucose can become fructose by moving that bond around as we begin our work here in this set of reactions. So you'll notice the enzyme here is called an isomerase, and all it does is it opens up this ring for a moment, it slides the ketone or the carbonyl over to the second carbon, and then recloses the ring. And so here is our fructose 6-phosphate. And notice the numbering system just like it was before, right? 1, 2, 3, 4, 5, and 6 is the one outside the ring that has the phosphate. Again, with the fructose, 1, 2, 3, 4, 5, 6. Carbon 1 also being outside the ring now because it's a ketose. All right, so again, we said in order to get the repulsion, we're going to need more than one phosphate on here. One phosphate is all fine and good. It's higher energy because we had to break down an ATP, but if I want the sugar to be activated, I need another phosphate. And so we're going to do the same thing we did on the first one, split an ATP and condense the inorganic phosphate with the alcohol, and here we go. So just like the first time, we're going to have the same set of other reactants. We've got an ATP that's been consumed and turned into an ADP, and again, we've got a water coming out because it's a condensation reaction. That'll put you on to page 2 of our worksheet packet. Now, at this point, activation phase is in full swing. We've put 2 phosphates on. We've spent some energy here. Remember, the end goal of metabolism will be to create a lot of ATP. Right now, we've been generally consuming it. But we've done what we really needed to do in order to get to this point. Our product is high energy, and it's ready for the dramatic change that we want because we've got negative charges at both ends.